Good morning, madam. My name is Mugilan. My matrix number is 23 DBT 20F 2007. I'm from DBT 3A. Today I'm going to brief about purification of fluorescent protein. In the simulation, we will purify protein expressed in E. coli cells from a recombinant plasmid in order to obtain the protein. So we should first lysis buffer. We should use lysis buffer to open the transformed E. coli cells. So for the process, we need many materials such as the E. coli EC1 solution, EC1 solution, then EC2 solution, and then the elution buffer, which is EB, then lysis buffer, which is LYB, and then the binding buffer BB, then wash buffer WB, column accumulation buffer CEB, and balancing tube, which is used for the microcentrifuge. Then for the micropipetting equipment, we need a P2 pipette, P20, P200, P1000. Then for the tick box for every pipette. Then for we need also empty tubes, which is labeled as RFB tube and super tube. Then other than that, we also need microcentrifuge and biohazardous waste and waste collection jar and timer, and then vortex and uh, chro- chromatography column. Okay, first and foremost, we have to pellet the cells. For that, we have to put the EC1 solution into the microcentrifuge. After that, we have to put the balancing tube inside. Then we have to put in put in the opposite side to check the balance. So centrifuge looks balanced and ready. Afterwards, close the microcentrifuge and put for 10 minutes. Afterwards, we have to take the tubes into the racks. Afterwards, we have to open the EC1 tube. Afterwards, we have to open the waste collection jar. Then we have to tilt this tube into inside. Then we already pellet the cell. For the second process, we have to pellet the additional cells. For that, we have to take the P1000 micrometer micro micro set the P1000 to 1000 microliter. For that, we have to add until 1000. Okay, we already got that volume, so save the volume. Afterwards, we have to attach the tip. Then open the EC second tube. Then we have to draw about 1000 microliters. Okay, we already got the samples. Dispense the culture into EC1. So our EC1 is already open. Okay, we already filled ready. Then open the biohazardous waste and eject the tip. Afterwards, close the lid. Then have to put the tube in the microcentrifuge for 10 minutes again. Afterwards, open the waste collection jar. Afterwards, tilt the solution inside. And our second pellet additional cells also done ready. Afterwards, we have to set the P200 to 150 microliters. Then we already got the 150 microliters, so save the volume. Afterwards, take a tip. Afterwards, open the EB tube. EB tube is over here, and open the EB tube and draw 150 microliters. So we already got the amount. Then dispense the culture into uh, EC1 culture. Then eject the tip into the biohazard waste container. Then we move to the fourth procedure. We have to lyse the cell. For that, we have vortex the EC1 tube for 10 seconds to resuspend the cell pellet. So we have to take the EC1 tube. Okay, then we have to put in the vortex. Afterwards, we have to attach the tip uh, tip for the P200. Open the LCB tube over here. Then we have to open the LCB tube and have to withdraw 150 microliters. So we already got that amount. Then so we have to dispense this amount of uh, lysis buffer into the EC1 solution. Afterwards, we have to open the biohazardous waste. Then we have to put the tip inside there. So we have to eject the tip. Then we have to eject the tip inside. Then we have to centrifuge the tube. Before that, we also have to put the balancing tube inside. Then centrifuge it for 10 minutes. Afterwards, I have to take the tube. So then we have to set time for 12 hours to incubate the solution. Then we have to start the timing. Let's move to the fifth procedure, which is equilibrate the column. So place the waste collection jar inside the under the chromatography column and open the column to drain the water. Afterwards, spin EC tube inside the centrifuge. After the centrifuge, the EC1 tube, we have to set the P200 into 200 microliters. Okay, and then save the volume. Then we have to open the EC1 solution. Then draw about 200 microliters of super Nanton. Then take the super tube. Then dispense the super tube in the super tube. Okay. Then open the biohazardous waste box. Then remove the tip. Afterwards, we have to close the super. Then let's we move to the sixth procedure. Afterwards, we have to open the BB solution. Then we have to attach a new tip. Then draw about 200 microliters. And afterwards, we have to dispense the BB into the super tube. Then we have to open the biohazardous waste box. Then have to eject the tip. Then let's we move to the seventh procedure. For this, we have to take the P1000 micropipette and the open the tip box. Afterwards, we have to take the tip and draw about 400 microliters of super solution. Okay, we already get the volume, so save the volume. Then the super solution over here. So we have to take that mount. Then dispense the red super solution into the chromatography column. Then we have to open the biohazardous waste box. Then we have to eject the tip. Then place the waste collection jar under the valve. Then open the valve. So afterwards, we going to move on the eighth procedure, which is wash the column. Wash the column is again set the pad to the 1000 microliters. We already got the volume, so save the volume. Then get a new tip. Then dispense of WB solution. WB solution is over here. So we're going to remove. Okay, then we have to take the mount. So we already got that mount. Okay, and close the lid. Then open the biohazardous waste box. Then dispense into the chromatography column. And finally, we have to open the column and allow the liquid to drain into the waste container and close the valve when the liquid is just above the resin bed. Then we already done the process. Then now we move to the last procedure, which is elute and collect the RFP protein, which is the first step is using a new tip and draw 1000 microliters of EB solution. So we're going to put the that one, and then we have to take the EB solution. Afterwards, dispense the EB solution into the chromatography column. Okay. Afterwards, open the column by turning the valve and watch the red protein traveling through the columns. When it nears the bottom, close the valve. Okay, done. After that, place the open RPF tube under the valve. Okay, under the valve. Then open the column to capture the RFP solution. Okay, and then we have to collect the RFP tube. Then we have to close the RFP tube. Then place the waste collection jar under the valve. Then dispense. Then we have to dispense 1000 microliters of column ECB solution into the chromatography column. Okay, then we already got that amount. Then we have to dispense this into the chromatography column. Okay. Then we have to dispense this. Then we have to repeat the process. Then we already done the process. Afterwards, then after purifying protein by column chromatography process, we get the results. For ECB1 and ECB2, is the color is red and slightly cloudy. This is because the bacteria replicated in LB or AMP or ARA and produced RFP, which remained inside the cells. And the predicted results is not same with the ideal results. Ideal results is for most the upper side is highly hydrophobic proteins. After that, the moderately hydrophobic proteins, and lastly we got the hydrophilic proteins.